So now that we've talked about the mechanisms of epigenetics and how it works, we can explore some cool phenomena. First being position effect variegation, which you've already seen. The second one is called gene imprinting. So position effect variegation, you guys have already seen this, so let's just go through it quickly. Basically the concept of depending on where a gene is located in low condensation or high levels of condensation will depend on what will change the level of expression. And this is illustrated really nicely with an experiment that was investigating eye color in flies. And what happened is they mutagenized a bunch of wild-type flies. And in one of the lines, they found that the wild-type copy of the red eye color locus was moved more closely to an area of high condensation. In an area of facultative condensation, where we see that in some cells it's a little bit less condensed, in some cells there's high level of condensation. And so this showcases really well how levels of condensation can be different between cells. So over here in this group of cells, we see that the wild-type copy is not highly condensed and it's free to be expressed. And in the white cells, levels of condensation do not allow for expression, and that gives to the variegated eye color. So that's position effect variegation. Let's talk now about a concept you haven't seen, gene imprinting. Concept that two gene copies can have different levels of condensation. And in particular, gene imprinting, where one allele will have levels of condensation depending on whether it was inherited from the mother or father. So having a look here in a sperm or an egg, we see that at the same locus, they have different epigenetic condensation. And in the child has one copy that is more or less condensed than the other. So let's look at a specific example, insulin-like growth factor, whereby the maternal gene in red here is transcriptionally silenced through epigenetic marks, whereas the paternal copy is expressed. And so when you have one copy of IGF-2 that is expressed and one silenced, you get normal levels of insulin-like growth factor and normal fetal development. But what happens if, for example, epigenetic marks from the maternal gene are removed or no longer present? What happens is both genes are expressed in a paternal fashion. You get overexpression of IGF-2, which affects fetal development and causes disease. Now, gene imprinting affects only a few genes in the genome, and a lot of them are related to growth and development. And there's an evolutionary theory behind why this phenomenon, gene imprinting, might exist. And it's the concept that males want every baby to grow as big and strong as possible, and females don't necessarily. Because the man does not have to raise and grow the infant inside their own body, give birth, and necessarily raise it, they want every sperm to produce a big, big, big baby. Whereas the mother, on the other hand, has some limitations. They cannot necessarily afford to have babies that are draining too many resources. For example, if the baby grows too big, they cannot physically give birth to it. And so this is represented genetically as the male genome tends to promote growth, and the female genome in a way limits growth. And so that's what we see with some of these gene imprinting genes. But like I said, this is not found in every type of gene. Gene imprinting is a relatively rare phenomenon, but it's interesting to think about why we might see these different levels of epigenetic marks. This also relates to why same-sex couples are unable to have children. For example, a lesbian couple would be unable to have or produce a child with two eggs. Let's say, for example, you took one with an X chromosome and one with another X chromosome. Why couldn't they make another female baby? One of the many reasons is that the epigenetic marks would not be complementary. As you can see here, if you have two maternal copies of this gene, you have improper growth. So this is one of the many reasons why you need a maternal and paternal sex cell together for proper fertilization and growth. And there's a lot of details here, and I won't expect you to memorize insulin-like growth factor. What we definitely want you to keep in mind is that maternal, or rather epigenetic marks can be inherited from the parents, that the alleles can have different levels of epigenetic marks. So just because a gene is at the same locus doesn't mean that its other copy will have the same level of condensation. And that level of condensation can correspond to the mother or father, for example. So keep that in mind. Now we've covered these two concepts, let's look into some of the applications of how epigenetics affects phenotype on a larger scale. Because what we've talked about before, or what you're introduced to earlier on, is that of course our genome has an important effect on what we look like, and so does the environment, this nature versus nurture concept. But where epigenetics really kind of blew the field away, it's a relatively new field, is that it's a bit of a combination between genetics and the environment. The level of epigenetic condensation, as we've seen before, changes expression and will have an effect on phenotype. But what also occurs is that cues from the environment can affect how condensed epigenetics is and can alter and add or remove epigenetic marks. The genome does this as well. And so the epigenome reflects both the environment and the genetic material within an individual. And a good example of this, here, let me just erase this, is given here. So let's run through this for a second. For example, this is drought stress in plants. And so if you take just a regular plant and dehydrate it, expose it to drought, it obviously is unhappy. If you do this to the point where you don't necessarily kill it and you can recuperate it, then it gets healthy again. What happens is if you expose that same plant to that same stress that it already experienced, it's more robust to the stress. And this is what's called stress memory, is that epigenetic marks have changed the genome in such a way that it's prepared for a stress that it's seen before. And so in this way, we could consider that plants remember what they've been exposed to. And so even in our own bodies, we remember the stresses and the experiences that we've had through our life through epigenetic marks, not just in memory. And what's interesting is if you repeat this experiment, you expose a plant to dehydration, then rehydrate it, and you take this plant's offspring and expose it to dehydration, it still shows a more robust level. It is more robust against that same stress as opposed to a normal plant. And so what we're seeing is that epigenetic marks, these epigenetic memory can be passed down through generations. This has implications for how well crops grow. This is obviously becoming more and more relevant as we want to train crops that can deal with climate change, different stretches such as drought, dehydration, hotter climates, salt water with sea levels rising. This is very active research field for training or getting the right crops that are more epigenetically flexible or prepared for different stresses. But epigenetics also plays a role in disease development, aging, and cancer. The levels of expression change throughout age, and when you get cancer and different diseases, just like we saw earlier with the gene imprinting, epigenetics plays a very important role in how genetics affects the phenotype and the outcomes of individuals. It's a relatively new and emerging research field. We've only known about this for the past couple decades. And if you're interested in this, there's a very cool course, Bio 347 Epigenetics, goes into all sorts of cool epigenetic mechanisms, how they work, and what we know as of right now. So if you're interested in this, I highly recommend the course. And like I said before, we've covered a lot in this epigenetics. This brings us to the end of the epigenetics content. We've covered a lot, but definitely make sure that you understand that epigenetics can be passed down through generations, it affects expression, and it allows cells to be different from each other.